Welcome, everyone. We'll take our seats. Welcome to those watching from afar. And it is my pleasure to introduce Dr. Gwenn Garden, who is here to give us a grand rounds lecture today. Dr. Garden is a professor of neurology in this department and is also co-director of the Center on Human Development and Disability. That's which is kind of a disease. So Dr. Garden is going to talk about some of her work here today on neuroinflammation, and it's all very exciting work. And Gwenn has also, of course, had a very important role in being a driving force to getting more research training for our residents, mentoring for those who want to pursue research after their residency, and setting up some infrastructure to expand our experiences beyond the clinical. So I will give you Dr. Garden. Thanks.
So the first test is, can I put this on, right? The second test is, can I make sure my hair doesn't provide feedback? Okay, so today I'm going to start, the talk's going to be in 3 parts. So for those of you who have somewhere to be, I'll let you know in the beginning that there are going to be 3 parts. The first part is going to be on it's going to be the most sort of basic and philosophical on the question of what actually are microglia. The second part is going to be more on some research being done in my lab on microRNAs and what they do in microglia. And the last part is a little bit more future-oriented and hypothetical about how we could use microglia for therapeutic development and also biomarker development. Okay, so on to our first sort of philosophical question. So for those of you who are from Seattle, and those of you who have moved to Seattle, what makes you a Seattleite?
Water repellent?
Water repellent, yes, okay.
Slow driving.
You don't jaywalk.
You don't jaywalk. I agree with that one.
Vitamin D deficiency.
Vitamin D deficiency, okay. So those are also things, and I love vitamin D deficiency because vitamin D deficiency implies that you've been here for a long time, right? So, okay, so I think, if you think about what makes you from somewhere, is it where you're born? Or is it the experience that you've had in that place, right? So most of you would probably agree that my daughter, who was born here in the city of Seattle, Swedish Hospital, sorry if there's anybody from OBGYN here, who was born here, is a Seattleite, all right? But what about me? So, I know we look so much alike, it's fun. It drives me crazy that sometimes she even wears her hair like that. Am I a Seattleite if I was not born here, but I moved here as a very young child? And that's a kind of question that comes up in biology sometimes. What makes a microglia a microglia? Does it matter where it was born? Or is it the experience that that cell has in the brain that defines whether or not it's a microglia? And I would posit today we're going to talk about this question, but I don't think we really know the answer. So where do microglia come from? We're going to talk a little bit about the history of this question. So in the 80s and 90s, when I was first learning about these cells, the answer was that microglia came from the bone marrow. And before the 80s and 90s, actually, people didn't think about it. Nobody wrote about it much. But in the 1980s, 1990s, basically the molecular markers that microglia seem to have suggested that they derived from bone marrow. They were like bone marrow macrophages. And then some experiments were done when you could first start knocking out genes, and they knocked out a transcription factor called PU1. And PU1 knockout mice had no macrophages and no microglia. So that was the answer. Microglia came from bone marrow. And people started doing bone marrow transplant experiments where they irradiated the mice. They got rid of all of the mouse myeloid or hematopoietic precursor cells. They irradiated the mice, and they replaced those mice with bone marrow that expressed green fluorescent protein. And those mice, lo and behold, had cells in the brain that looked like microglia and were green. So proof, microglia come from the bone marrow. How many of you believe that?
Your body language suggests it's not true.
Okay. That's a great question. So, okay, so that was the 80s and 90s. That was really when I started thinking about studying microglia. I thought they came from bone marrow. And then in the 2000s, people started doing some experiments that suggested otherwise. So first of all, we got more sophisticated molecular tools, and we started looking more generally using profiling experiments at gene expression microglia, and they really did seem to have some unique gene expression patterns that were different from other types of macrophages, especially circulating macrophages. People even started to be able to separate macrophages in the brain that lived in the perivascular spaces, the choroid, etc., from microglia based on the level of expression of specific markers, and one of them was CD45. And then some experiments were done using a couple of models. One was something called parabiosis. Anyone know what parabiosis is?
Is that where you glue them together?
Exactly. So they took two animals, one expressing this green fluorescent protein in its cells and one not, and they actually sutured their vascular systems together, and the animals lived. And I don't know if you could still do those experiments with current high-cook standards, but they did this, and the green cells occupied many different organs in the animals that they were sharing vascular systems with, but they didn't enter the brain. So parabiosis suggested that bone marrow-derived cells, if they really do go to the brain, it would be under very rare circumstances. And then people went back and they started doing these bone marrow transplant experiments again. And instead of doing them the way that they had always done them, where they irradiate the animal, get rid of its endogenous host immune system, and then put in the graft, they actually shielded the head with lead so that the head of the animal would not have radiation. So the endogenous microglia remained. Then they put in the green bone marrow, and those green bone marrow cells also very rarely crossed into the CNS where the head had not been irradiated. So that led people to think that, again, microglia are not derived from bone marrow. And, again, I would say that I've seen data where those experiments have been redone. It's not quite as clear-cut, but still the suggestion that most microglia are probably not coming from bone marrow.
So how do they explain the original studies?
Well, the hypothesis is that radiating the head causes injury, breakdown of the blood-brain barrier, and actually attracts those bone marrow-derived cells into the brain. And then once they're in the brain, under the influence of the environment, they start to look like microglia. But that the endogenous microglia that most people have who have not had irradiation to their head would not have come from the bone marrow. So, okay. So now, we're in the 2010s. Sometimes I feel like it's hard to believe we're in the 2010s, but we are. And some experiments done around 2010 suggested that during embryonic development, microglia actually don't come from the bone marrow at all, not even during development. They come from the yolk sac. How many of you knew that we have cells in our bodies that actually are derived from our yolk sac? Neffy knew. Okay. Yeah. So, but I did. When I saw that paper, I was like, really? I didn't know. I didn't know we actually have that. But there are long-lived cells that are yolk sac derived that enter the brain during embryonic development, and it turns out that in the case of microglia, not only do they enter the brain from the yolk sac, they enter and the whole CNS. They enter the whole CNS from the yolk sac before we even have a beating heart. Okay, so you can see, I have to remind myself not to point with my finger. You can see here, these are in the white bars are the microglia, and they are entering the CNS of the mouse. These are some marker experiments done. And so the white bars are showing how many microglia are entering the mouse before, here's where the heart actually starts beating. So microglia are born in the yolk sac. They enter the brain very, very early on in the development of the embryo. Around the same time, people started doing experiments where they would have one cell type be green and another cell type be red. And they were able to see that if you use a driver promoter for the molecule CX3CR1, which is a receptor for a chemokine, that those cells, when those cells are green, those are microglia, and cells that express a different chemokine receptor called CCR2, which they labeled with red, those cells would stay out of the brain parenchyma, and those cells were derived from bone marrow. So in the brain, you have bone marrow-derived macrophages, which express CCR2, and yolk sac-derived microglia, which express CX3CR1. And the two populations in the normal animal really didn't intermix. So here's the diagram of how the experiments done around 2010 suggested that microglia developed. So here's the yolk sac, and you have these cells which are, they're macrophages because they don't require this transcription factor, and they enter the brain and become microglia. And then you have from the bone marrow cells that still don't require this transcription factor. They circulate around the bloodstream as what's called a monocyte, and they eventually enter the brain and become choroid plexus macrophages, perivascular macrophages, and laryngeal macrophages. So I think some people kind of thought, oh, we figured it out. It's defined now. Microglia come from the yolk sac, and all the other brain macrophages come from the bone marrow. But there was one experiment that suggested there may be some variation from that theme. And this experiment looked at the transcription factor called HOXB8. And HOXB8 is a transcription factor that's expressed in a subset of monocytes. And it looked like from this experiment that if you looked at cells that were expressing HOXB8, this is a P2 animal mouse brain, and there's a small subset of microglia-like cells that are expressing green under this HOXB8 transcription factor. And as overdevelopment, so here's P2, here's P14, P90, this population expands. So when the animal's born, it really doesn't have many of these cells that look like microglia, but over development, you get more of them. And if you remove these cells, so using a genetic trick to get rid of all the HOXB8 expressing precursors, the animals had a very small but significant reduction in the total number of microglia. But interestingly, they developed, my arrow is on my screen, but it's not on your screen. Okay, I don't know how to make that work. But anyway, so if you, really, it's on my screen, but I can't see it. So if you look at that small reduction, you think, well, what difference does it make? It's bad. Okay, so what difference does it make? You think not much, but actually these animals were phenotypically quite distinct. They developed what the researchers called obsessive compulsive disorder because they were chronically removing their own fur. And that was quite a distinct phenotype for these animals that are just missing this very small portion of their microglia. So there may be more than one origin of microglia, and it may be important that you have the full composite of all the different types of microglia. So what about those microglia that you're born with, or at least that mice are born with? Are they the same ones that are present throughout the whole entire life of the organism. So, I'm not sure how well this is going to project, but I'm going to talk you through it. Quite recently, 2014, a paper came out that suggested that actually not only that, well, we don't know how long microglia live in the organism's lifespan, but if they don't live the whole lifespan of the organism, there is a population of progenitor cells that can replace them. And up until 2014, no one really knew that these cells existed. So this experiment, what they did was they gave animals a drug that's Mark knows well from a company called Plexicon, that is an inhibitor of a molecule called CSF1 receptor, right? So microglia depend for their survival on a molecule called CSF1. And if you block that receptor, microglia die. Actually, all myeloid cells die, but microglia also die. So they gave these animals this drug and blocked their CSF1 receptor, and the microglia died. And then after they stopped giving the drug, they watched what happened to the animals. And over time, they started to develop new microglia. And these microglia were dividing. So here's a marker of cell division, P67, and you can see there are dividing cells. And this increases over time, and you can see, not very well here, but there's a lot more red here for the P67 marker, and eventually you have a lot more green microglia. And this is quantified here in the graphs below in 3 different regions of the brain. So here the green is the microglia, the P67 is in the blue, and the white is cells that are labeled with both, right? So by day 3, after you stopped giving this drug, you had an increase in the size of the population of cells that were expressing the microglia marker and P67, and that occurred in hippocampus cortex and piriform cortex, pretty much everywhere throughout the brain. So microglia, if you remove them, can be reborn, and it actually happens very quickly within 3 days. The full repopulation probably takes a little longer, but by 3 days you have quite a lot of new microglia.
Do microglia ever form neoplasms?
Well, yeah, we don't know. Not that we know for sure. Mark says no. But I don't know if I completely agree with you, and I'm going to tell you why in just one question, in one slide here. Yeah.
Does this imply that perhaps the yolk cell derivatives in a microglia are actually stem cells?
Sorry.
Yes. So interestingly enough, these new microglia, before they become microglia, express nestin. So nestin is known as a stem cell marker, and what they did was they labeled these recovering animals. They were expressing GFP under a microglia promoter. They labeled them immunologically for nestin and for this proliferation marker Ki67. And they found that some cells, you can see one here that's kind of highlighted by their green horizontal lines, express Ki67 and nestin at the same time. First you see them where they don't have microglia markers, they're just nestin and Ki67 positive, but then you do see cells that are also Ki67, nestin, and CX3CR1 positive. So these experiments suggest that these cells are stem cell derived, nestin positive, start to proliferate, and then become microglia. This is normal mouse, right? So I'm going to tell you that it didn't happen quite this way, but if we ask the question, can we find these cells in the
Can
brain?
I ask, Gwenn, before you leave that interesting observation, do we know whether these are from the yolk sac or whether these are those few microglia that slip in from bone?
We don't know the answer to that at all yet, but I'd like to answer that question myself, but we don't know. so the first thing that, we thought was, well, could we isolate these stem cells? And like I said, didn't really happen this way, but I'm going to present it like it did. So we wanted to isolate microglia in culture from adult mice and see if we could find these stem cells. And so we decided to perfuse animals, dissociate the whole brain, and then use what's called magnetic bead isolation for a common myeloid marker called CD11b, which is the receptor for CR3, which is a molecule in the complement. And it just so happens that you can buy these beads that are pre-labeled with CD11b, and we use them when we do flow cytometry experiments, which I'll show you some later. And so we did this, and we isolated the cells that had CD11b, and then we put them in culture with MCSF, or CSF1, which we're talking about, and we cultured them. And eventually, over time, we got cells which looked like microglia. So I'm going to see if I can... Still not happening. Okay, maybe if I go like this, I can get... I'm getting a laser pointer here.
Yeah.
There we go. Okay, so you can see cells that look like microglia. All right, and they label, in this case, they're labeled with a molecule called, antibody to a molecule called IBA1, which is again another common myeloid marker. So then, what really happened was we were doing this, and then we saw the paper about these microglia stem cells, and then even I didn't immediately think of it until I was at a meeting talking about this new method of culture that we developed, and someone said, "How do you know you're not culturing these stem cells?" I just happened to be talking right after the author of that paper. So I came back and suggested this to a postdoc in the lab, and she immediately went and stained these cultures for nestin. So if you culture the cells for short periods of time, here's 1 week, and you label them with nestin in green, pretty much all of the cells are nestin positive, a few are nestin negative. And if you wait in time, eventually they start to become more cells which are IBA1 positive. There's still some nestin positive cells, and there's some cells which are positive for both markers. So we're sort of recapitulating this development from a progenitor to a microglia in vitro by starting with CD11b positive cells. I have to tell you, it's one of the few times in my career that I almost didn't believe results that were really so obvious because it seemed so unlikely, but we did this several different ways, several different tools that we have available, and it's really quite convincing that actually these nestin positive cells are there and that they eventually start to express microglia markers with time and culture.
Gwen, I can't help but ask the question. People have been making microglia cultures for years.
Right.
Did they just not stain with nestin?
So we've stained the neonatal microglia cultures with nestin, and they don't express nestin. So it's something quite specific to the isolation of the adult population. So what happens when you make microglia cultures, this is getting kind of technical, but when you make microglia cultures from neonatal animals, you essentially culture all the cells of the brain together, and then you take the cells which float off those cultures and you plate them secondarily, and you say, and then those cells tended to have microglia markers, and we say those are microglia.
But they're not divided.
And they don't divide in culture, those neonatal microglia.
When nestin isn't the only stain for stem cells.
Right.
And many people argue that many cells that are stem cells in fact are nestin negative. And
Right.
there's a huge subpopulation where the cells get positive or have another marker, CD133.
We haven't looked at CD133 yet. We have looked at GFAP, and they actually are GFAP positive. CD11b positive, GFAP positive, nestin positive. So here's a sort of a panel of things that we have looked at. So on the white bars are microglia that we isolate from the brain, the adult brain, that are CD45 intermediate. So this is just a way of saying these are the parenchymal microglia. So we use markers to say that. And we separate out all the perivascular macrophages. Parenchymal microglia, we made RNA. So that's the white bar. And then the black bar is these cells that are grown in culture for 7 days. What you can see is that, these have been defined by many other labs as microglia markers. So hexosaminidase B, TREM2, CX3CR1, I told you about that one already. And CCR2, which is a macrophage marker, it's very low level expression. You can see these numbers are quite small. But there are probably some cells in that culture that express CCR2. Here's CSF1, it's the ligand that microglia depend on for their survival. Here's the receptor. Here's GFAP, nestin, and then IBA1, which we've talked about. So the cultured cells are expressing GFAP. They express GFAP, mRNA, and protein, nestin, and then c-KIT, which is another stem cell marker. So we haven't looked at CD, but these are the three that we've looked at so far. All right? Mark's smiling now. And interestingly, if you then take these cultures and you expose them to interferon gamma, we need to do more different experiments like this, but so far, this is the one we've done, you can see that they actually are able to differentiate into microglia more quickly. So after 24-hour exposure to interferon gamma, you start to get expression of hexosaminidase B, CSF1R, and IBA1, and nestin expression decreases quite dramatically. So this is one key piece of information that suggests that these progenitor cells, one of the things that probably promotes their differentiation into microglia, stops them from being progenitor in their identity, is a pro-inflammatory stimulus. Okay, so this is the end of the first part of the talk. So, I think the summary that I kind of come from is that microglia come from several sources. Some come from the yolk sac, some come from bone marrow, and in the adult brain, some are probably derived from this progenitor population that we really don't understand much about yet. So there's this population that migrates from the yolk sac, there's the HOXB8 population, and then the adult progenitor population. Okay, and then these adult progenitors, you can get them to differentiate by treating them with a pro-inflammatory stimulus. So this is actually, I would say, the most interesting question that I've been thinking about for a really long time in my scientific career. What does the existence of these progenitor cells imply? Somebody already talked about tumors, right? Here is a second nestin-positive population of cells in the brain. Who knows what happens to them? Yeah. GFAP positive, nestin positive, yeah.
So the thing is, if you go back, you have very few nesting positive cells when you first put the cells into culture. Now either you're losing everything that isn't nesting positive, or the nestin is getting turned on in culture, and what you're assuming is a progenitor cell because you're present in the nestin is actually a cell that has turned nestin on.
So, okay, so I'm going to repeat the question because it's a really good question. So the question is, because I have people listening in Harborview. So the question is what's happening that we're turning nestin on by our, in culture, by our experimental manipulations in culture? And I think there's some degree to which that's possible, but I think mostly what we're doing is losing most of the nestin negative cells because the yield of this kind of culture is very low. So, we get back, maybe 5% of the cells that we put in, right? So the second answer to that question, that I don't think that we're inducing nestin expression per se, is because it happens in vivo, right? If you kill off the microglia by blocking CSF1 signaling, then these nestin positive cells start dividing. So the real question is, are there nestin positive cells living in the brain that you can identify without doing any kind of experimental manipulation? And I'm in the process of writing a grant to try to do that, but I don't know the answer yet. So, but yes, but so let's just say, for the hypothetical, maybe we haven't 100% proven that these cells exist, but what could we do with this progenitor population? And then why do we have them? Right? I mean, do we need to replace microglia? Do microglia die over time? What are the stimulus for these progenitor cells to start dividing and differentiating into microglia? We've already, I think, identified that pro-inflammatory cytokine interferon gamma can promote differentiation, but we don't know really, other than the killing off of all the other microglia, what causes them to divide. Placing them in culture where there's a very low density probably induces them to divide. But what other circumstances? We don't know that. This is, I think, a really important question. Is there some sort of replicative senescence? If we ask the question, why do we accumulate misfolded protein in the brain with age? Okay? Maybe our defense mechanisms include microglia. As microglia internalize misfolded protein, eventually they become activated, they may die, and then the turnover that's stimulated by that may have some time limitation. Maybe they can only go through so many cycles of cell division, and then you don't have effective means of removing those proteins. Very theoretical. I said the beginning will be philosophical. And then the next question, leading into the second part of this talk, is, does experience influence the eventual inflammatory profile of the microglia that are born of progenitors that have been stimulated by inflammation in the past? And what I mean by that, or what is the term for that, is something called epigenetic influence. So in the beginning, I asked, what makes you a Seattleite? Is it, are you born here or your experience in Seattle? And we could say, for many of us who've lived here for a long time, we've been very influenced by this environment. Those of you who lived here for a long time, when you see that picture of the beautiful sunset over, Pike Place Market, it causes an emotional, almost a hormonal response that you wouldn't have if you hadn't lived here, right? And I don't know if that's mediated by some epigenetic change, but experiences do have lasting impact on ourselves, and we call that epigenetics. So you can take a narrow perspective on what's epigenetic or a broader one. There's a lot of things that can be included under epigenetics if you take a very broad perspective. And a broad definition is the modulation of gene expression without altering your DNA. So this is a little bit more narrow. Some people say this is specific chromatin modifications which are able to persist with cell division. And there's some very new work, which I didn't have time to review for this talk, about methylation patterns in microglia. And this is, I think, going to be a very important question over the next few years to answer. Are these progenitor cells becoming methylated by experience? Let's say you have early head trauma. Does that cause replicative senescence, and so you have more risk for Alzheimer's disease? Or does it cause some sort of epigenetic change in the progenitor that is transmitted into later born microglia that then change that inflammatory response and make you more likely to have Alzheimer's disease? Then there's this whole new area of non-coding RNA. How many people have heard about non-coding RNAs? A few more. Okay. So for those of you who haven't heard about non-coding RNAs, there's at least two different kinds, and there probably are more. So, we have this this huge genome, and most of it doesn't become messenger RNA and protein. Most of it's just there, and we don't know its purpose. And some of those regions that we don't really know the purpose actually do make RNA, but those RNAs don't have the particular sequences that enable them to become proteins. Some of them are long, non-coding RNAs, and they actually interact with other RNAs, form double-stranded RNAs, do things to regulate gene expression that way. And some of them are short, and they've been called microRNAs. And microRNAs are a really interesting class of regulatory molecules, in part because they are so small. And because of their small size, they become very interesting to people as potential therapeutic targets. So, so what are microRNAs? So basically what happens is RNA is transcribed. It's usually a fairly long piece of RNA becomes what's called a pre-microRNA. Then it gets exported into the cytoplasm. It's cut up by a molecule called dicer, and then it interacts with this protein complex called the RISC complex to target messenger RNAs. And when a microRNA binds by complementary sequence with a 3' region of a messenger RNA, it can do two things. It either causes the message to not be translated, or it can promote, because of the reaction to the double-stranded sequence, the degradation of that mRNA. So these small RNAs, microRNAs, are negative regulators of gene expression. They're obligate negative regulators. Okay. So microRNAs have already been shown to have very important roles in development and in inflammation. And this is just a profiling experiment that was done by another neuroinflammation researcher, where they took microglia, the neonatal kind that we commonly use, the ones that are grown in culture, and they exposed them to either lipopolysaccharide, which is a pro-inflammatory stimulus, it's a bacterial wall molecule, or to interleukin-4, which is supposed to promote anti-inflammation. And the pro-inflammatory stimulus caused the change of microRNA expression, as did the anti-inflammatory stimulus, and they identified the ones that were most predominantly changed in expression. I'm just going to show you some that we're going to talk about here. It's microRNA 155, which is one that we've been very interested in the lab. MicroRNA 124, which is fascinating because it's important. So microRNA 124, you need to keep microglia in their normal quiescent state, doing what they normally should do. It gets decreased in inflammation, whether it's the pro-inflammatory or the tissue repair phase of inflammation. And then microRNA 145, we'll talk about that a little bit later too. So microRNAs are important because if you think about what a microglia has to do, and this is, I kind of started thinking about this before we even knew that microglia could kind of die and be reborn again, right? If you think about all things that microglia have to do, they have to be able to change, right? And microRNAs are known to help cells change in a sort of a long-term way because they're very stable, small pieces of RNA. They can make change happen for hours, days, maybe even weeks, just by the change in their synthesis, right? So if you have an inflammatory stimulus, you want to change, but you want to change short-term. You don't want to completely change. Maybe you can have a 2-day, real strong pro-inflammatory phenotype, and then that you have a change in microRNA expression, and then you could go to a more of a tissue repair phenotype. So the overarching way that I was thinking about this was that there should be these phases. Microglia, in macrophage biology, they like to talk about polarization, but maybe less so now. But they used to talk about polarization. You have this type of macrophage and that type of macrophage. But microglia, maybe they just go through these phases. And so the idea was that they would initially respond to an acute injury, traumatic brain injury, stroke, infection, with these things that would call for help from outside, call the perivascular macrophages in, call neutrophils in, and have an acute inflammatory response. Then after that all happened and you needed to make it stop, right? Because if it kept going on, then you'd have brain injury that would persist. So then there'd be a second phase where you have signals that would cause it to deactivate. And then you'd have a third phase where you'd try to cause tissue repair. In tissue repair, there are signals, again, you'd signal to probably perivascular macrophages to help you come in, create new extracellular matrix. Maybe microglia actually help create new extracellular matrix, but they certainly secrete factors that promote vascularization. So revascularization, rebuilding of capillaries in areas of repairing from ischemic or traumatic brain injury. So what controls that phase switching? And that was something that we've been interested in for quite some time. And we started by thinking about stress response programs and reactive oxygen species sensors. And we came across this molecule P53, which anybody who's interested in cancer thinks about as a molecule that's involved in preventing cell division or even causing cell death. And in microglia, they activate P53 in response to pro-inflammatory stimuli, but it doesn't cause apoptosis. Maybe it prevents progenitor cell division. We never really thought about that, but they activate P53 even when they're in neonatal culture where they're not going to divide anyway. And that P53 activation appeared to also be important in vivo because if you look at stroke in a P53 deficient animal, after stroke, very much earlier than in wild type animals, you see this accumulation of microglia that express a molecule associated with the tissue repair phase. So P53, through this and several other experiments, we realized was important for promoting pro-inflammatory phase and preventing the moving into the tissue repair phase. So the question was, how did that happen? And through a whole bunch of microarray experiments and gene profiling experiments, we came across this transcription factor called C-Maf. And C-Maf is a transcription factor that was known to promote anti-inflammatory behaviors in macrophages. So the real question is, how does C-Maf regulate, how does P53 regulate C-Maf? Let me skip over that because I'm going to run too long. Because P53 is a positive regulator, turns things on, but it was suppressing C-Maf. So then the question became, is this through a microRNA? So here's this diagrammed out. The idea is you have DNA damage, reactive oxygen species, you induce P53, it induces a microRNA, and then that suppresses C-Maf. So we looked at this microRNA 155 because a paper had shown that it suppresses C-Maf, and we saw that there's a big role of P53. So in the absence of P53, you really don't get much induction of microRNA 155 in response to a typical pro-inflammatory stimulus. And if you look in mice that are deficient in microRNA 155, both in vitro, so these are these neonatal microglia. In the absence of 155, you have much more C-Maf, and in adult microglia that we extract by flow cytometry, also much more C-Maf. So microRNA C155 seems to be important. We also found two other microRNAs that are important by looking at this transcription factor TWIS2, which we know also promotes the expression of C-Maf. TWIS2 is a target for two known P53 dependent microRNAs, 34A and 145. So we have data for that. So this was kind of our model. The idea is you have P53 activation. It promotes these three different microRNAs, which then suppress C-Maf. So all this was done, pretty much using in vitro experiments, and all with mice. And the question is, are these microRNAs important at all for human disease? And this paper that came out suggesting that they are. So if you look at Alzheimer's disease brain, you see that Alzheimer's patients have a big increase in miR34A as well as in 155. And interestingly, in this paper, they also showed that microRNA 34A is a microRNA that suppresses TREM2, which is a molecule in microglia that also with the loss of TREM2, you get neurodegeneration in a recessive disorder, but in individuals who have one copy of that dysfunctional TREM2, they have a predisposition for Alzheimer's disease. So lastly, I'm going to just wrap this part of the talk up by saying that we used a short-term ischemia model and looked at the microglia 3 days later. So this is what happens. You get an increase in microglia after short-term ischemia ipsilaterally. And if you take those microglia out of the brain and you look at their microRNA 155, they have a big increase 3 days later, and they have a decrease in the expression of C-Maf. So I'm going to, again, skip over this part because I don't want to keep you here too long. And most of this is just technical bragging about stuff that we were able to do that I was surprised we were able to do. And so this is kind of our model when it comes to the ischemia response. So even though we think this is probably important and could be very important for more chronic types of inflammatory responses, because we can see this microRNA 34A at later times, in ischemia, acutely, it appears to be dominantly mediated by miR-155. Okay. Uh-huh.
Is the transcription factor C-Maf, is that a consistent finding across all macrophage lineages in promoting polarization to M1?
To M2.
M2 phenotype?
Yeah. So yes. So, okay, microglia behavior, the pro-inflammatory behavior is supported by P53. P53 appears to work by suppressing C-Maf, and it does this through microRNAs, which suppress another anti-inflammatory transcription factor, TWIS2, as well as C-Maf. And that we can actually show that this is important in an in vivo model. Okay, so lastly, is in neuroinflammation a therapeutic target? So neuroinflammation is common to pretty much every neurologic disorder that we encounter in the hospital. I'm hard-pressed to think of one that doesn't have it. Someone could probably think of one, but they're not many. There's lots of evidence to suggest that that inflammatory response actually contributes to pathology in a lot of degenerative diseases. So, maybe inflammation may not be important to the pathophysiology of epilepsy. I would argue that we don't know that it really could be. But in ALS, in Alzheimer's disease, in Parkinson's disease, stroke, we think it's important to the pathophysiology. And there are examples, such as HIV-induced neurocognitive disorder, where inflammation is probably the primary cause. In MS, inflammation is probably the primary cause of the neurodegeneration. So it could be very important. And does our relatively new knowledge about the unique origin of microglia enable us to think about ways of developing therapeutics that would have less systemic toxicity? To this point, things that, I should admit the things that we're working on are probably not going to be specific to microglia. P53 is really much more of a regulator in microglia than other macrophages, but C-Maf not so much, and these other microRNAs clearly are important in other macrophages. But just developing this way of thinking about microglia and ability to study them might make us able to find things that are specific to CNS inflammation. And are these microglia progenitors a potential target for therapy? So I like to ask this question recently, where we say, in the future, where we have personalized medicine, how will we know if our targeted therapy is working? Well, one way that we could know the answer to this question is if we can follow the inflammatory response. So whether or not inflammation is involved in pathophysiology, if it's a marker of the pathophysiology, we could actually potentially use it as a biomarker. And biomarkers are important for the development of new therapies and personalized therapies because in neurology, functional outcomes change very slowly. It's very difficult to do clinical trials because we are following our patients for such long periods of time to see any kind of change. Maybe not in GBM, but most other types of neurologic disorders progress slowly. So biomarkers would help. They help in more ways than just speeding the clinical trials by providing a surrogate outcome, but they also help people who are designing trials narrow the cohort to the individuals who are going to be able to be followed by that biomarker. And then if you narrow those cohorts, that can help to bring new trials to approval faster by developing these personalized indications that are validated by biomarker. So if you use MRI as a biomarker, which is really for the last 20 years what we've been using, maybe longer, it limits what you can do because first of all, MRI, the resolution isn't that great. It has limited molecular specificity. And in our patients, our adult patients with other age-related change, the outcomes that we're looking for at MRI can be confounded by other age-related pathologies that may not be targeted by treatment. So we need molecular biomarkers. And I would argue we need neuroinflammation biomarkers. So one of the reasons I'm encouraged by this is by this new technique called TSPO PET. And TSPO is a molecule that is dramatically upregulated in microglia in response to inflammation. And that a ligand that you radiolabel and do PET imaging that binds to TSPO can be used to measure inflammation. And so this is just an example in an animal, but this has been done in patients where you induce inflammation with a stroke and you see increased radioligand binding for TSPO. So we wanted to use this in some of our animal studies, but the question was actually using this model that we have where we do a very brief ischemic insult, which doesn't really cause a stroke but does cause neuroinflammation. Would there be a TSPO PET signal? And so that's the first question. And the second question, is that signal actually specific to microglia? Because there's some literature in the animal world that suggests that this TSPO molecule isn't expressed only by microglia. So we're moving forward anyway, and we used this inflammation molecule, this inflammation model that really came from John Weinstein's lab. And we isolated the cells, and we found that if we isolated microglia on the side of the stroke, we did see an increase in TSPO. We could see that by RNA, we could see that by protein. It didn't go up in astrocytes. And if we did radioligand binding studies with Nephistella, we saw that it was actually increased only in microglia. So the last part of the talk, the summary is that PET imaging for this molecule TSPO can be a biomarker of even very subtle types of neuroinflammation where we have limited neural injury, including this 15-minute MCAO reperfusion model. That increased TSPO is observed predominantly in microglia, and that the radioligand binding is limited primarily to microglia. So lastly, I just want to mention the people who really did this work. Currently in the lab, Macarena Aloy is a graduate student who's working on a MicroRNA project. Stephanie Hopkins is the person who really runs the lab and has done many of the experiments that I showed today. And Wei Su is a postdoc in the lab who's really taken on this microglia progenitor cell project and made it happen. And then I also have to acknowledge Sumi Jayadev, who many years ago when she was working with us really got much of this project started, and John Weinstein, who brought the stroke model to our lab. Jeff Chamberlain and Guy Odom, who enabled us to be able to manipulate microglia with AAV, and do some of the experiments that have proven to us that these really are this AAV system enables us to really molecularly modify these progenitor cell population, which we wouldn't be able to do without that, without AAV. And then Nefi Stella, who helped with the radioligand binding. So I'm happy to take any questions.
So Gwen, that was terrific. Don't take this wrong, but I have a little feeling that what was old is suddenly new again. I'm thinking about George Kreutzberg's model of looking at the brain after a fifth nerve injury in a mouse, and then finding that there was an enormous amount of cell division that occurred in the fifth nerve nucleus.
I'm looking for it. Is it up there? Oh, good. Okay.
And that these cells were primarily microglial cells. So do you think that that's just an example of these, resident, stem cells dividing? There's plenty of instances, I think, of very subtle things, well away from the brain, where you wouldn't interfere with blood-brain barrier, where you suddenly get a wave of microglial proliferation.
I think that it's okay. So the question is, if you think back to old models of microglia activation, primarily the facial nerve injury model, which really was the basis. Yeah, the basis of thinking about microglia activation for many years. Are those results due to progenitor cell division? And really, there was no way to interpret those studies at that time in this way. But now, knowing what we know, and this is putting together, the unpublished stuff that we have done with the one paper that has been published, I would say it's possible. And yeah, it would be great to go back and redo those experiments with new markers and try to see, do we recruit nestin positive cells to divide, and that those cells then become microglia. Tom?
Two questions. One, I don't think I've ever in this talk heard you use the phrase activated microglia. You see it all the time in the literature. Can you define what they're talking about when they say that?
So my graduate student's smiling back there because it's become such a, I think I've been beaten down and I don't use it anymore because it's become such a hot button term in the field, because it is so hard to define what is really meant by activated microglia. So as we're talking about the facial nerve model, so if you injure the facial nerve and you look in the facial nerve nucleus, you saw microglia change in their morphology, and that was what we decided meant activation of microglia, because they went from being these very ramified, thin processed cells to looking amoeboid and having a very different pathology. So that then went into culture experiments, this neonatal culture model where you would expose them to injuries, or this lipopolysaccharide, and they would express pro-inflammatory molecules, and that was called activation. And then as the microglia tend to follow the macrophage literature. The macrophage literature went to, well, actually there's more types of activation than just pro-inflammatory activation, and so then microglia activation started to encompass, other things in the macrophage literature, and then a bunch of old microglia people said, oh no, microglia are nothing like macrophages and you can't use that terminology. So I guess I try to keep it politically correct in the world of microglia. So that's why.
And the other question is, can you make human microglia from iPS cells?
So Sumi, do you want to answer that question?
Yes.
Yeah. So Sumi, in Sumi's lab, they are making microglia from iPS cells, yes. But it's very, I don't know, we might have to take you aside and have you, sign a confidentiality agreement.
Yeah, so you can, so there are, certainly you can make them from mouse embryonic stem cells, and there's a lot of people how to make it from human embryonic stem cells and human-induced pluripotent stem cells. And then the question then becomes, how are you going to decide if they are the same cell type that you've defined previously using other models of what a microglia is? What are the protein and genes expressed that are required before you say this is microglia?
So in case anyone is still listening at Harborview, the conversation is about what makes an induced pluripotent stem cell derived microglia a microglia. And it's, I have that on the top of my slides, what makes a microglia, and that's a big question right now. How do you decide this is a microglia once you start with the pluripotent stem cell?
These people who work in the stem cell field define stem cells usually by their ability to form neurospheres, and that's a fairly characteristic across all organs. Are you able to do a similar functional experiment with microglia?
So again, going back to like old studies, so if you form neurospheres, and Kevin's like waiting to talk, if you form neurospheres, you get microglia, right? That's always been the case. And we've never really understood exactly, so you take neuroprogenitor cells, you form neurospheres, and you get microglia. I don't know what that means. And then can we do it with our cells in culture? I don't think we've been doing this long enough to know the answer to that question. Yeah, Kevin.
Well, my question was fairly similar. I was just going to say, in your culture conditions, if you have cells that express nestin and express DNAB and express CK, do you think that they're still lineage restricted to microglia?
Well, they also express CD11B, right? That's how they got there in the first place. So I don't know the answer. Because I don't think, although I don't know.
Are they astrocyte wannabe?
Right, exactly.
Or then do those progenitor cells always only differentiate into microglia?
Yeah.
Would you?
We still don't know the answer to this. I mean, this is all like this stem cell stuff is less than a year old. So I don't. We have no funding. So we're still really in very early stages of trying to decide what this actually means.
Gwen, that's terrific. Thank you so much.